Well, good afternoon, everyone, and I think we may have some folks online as well. So I'm David Russimano. I'm the provost here at the University of Memphis, and it really is my distinct honor and pleasure to introduce our distinguished speaker, Dr. Mangilal Agarwal. I'll read his bio here in just a moment, just so you get kind of that formal introduction. But Mangi and I had the opportunity to work together for 13 years at IUPUI. And I first met Mangi when he was working in our Office of Research, working on enabling opportunities for faculty to collaborate, and was actually quite successful in his own right. And we talked about, is there a way to transition him to a tenure track role in the Purdue School of Engineering and Technology at IUPUI at the time. And I'm delighted to say that that happened. And Mangi was tremendously successful in his own right as a PI, and ascended through the ranks, became a full professor, a tenured full professor, and has reached even greater heights, I think, of excellence in his work. So with that, let me go ahead and read his formal bio to introduce him. So Dr. Agarwal is a professor of biomedical engineering and informatics and director of the Integrated Nanosystems Development Institute. The acronym is INDI, at Indiana University, Indianapolis, which was formerly a part of IUPUI. INDI is dedicated to innovative research and education in its focus areas of biomedical and energy applications involving nanomaterials, characterization, nanofabrication, and provides an institute for enabling several research programs by bringing together more than 30 faculty members with over 25 million in funding in just the last 5 years. After earning his PhD in engineering from Louisiana Tech, Dr. Agarwal was a postdoctoral research associate at the Institute for Micromanufacturing. He then joined then IUPUI in 2009 as the associate director for research development in the Office of the Vice Chancellor for Research, and later transitioned to a tenure track faculty position in the Purdue School of Engineering and Technology, and ascended to a tenured full professor of mechanical engineering and director of INDI. Besides enabling the success of others, Dr. Agarwal has received substantial funding as PI, including support from the American Cancer Society, National Science Foundation, NIH, DOD, Veterans Administration, and industry. And I heard you just were awarded NIH R01 to go along with your extensive portfolio of research. So please join me in welcoming Dr. Agarwal to the University of Memphis.
Thank you, Provost Russimano. That was a nice introduction, and I'm very happy, and it's my pleasure to be here and give you some research projects that we've been working on, and open for collaborations in future center type of grants as opportunity permits. So the topic of my talk today is multidisciplinary research from biomarker discovery to sensor development for non-invasive health monitoring. And I'll also cover some of the other applications in the same uh, area. So canines, dogs, we have seen them at the airport. We know that they can be trained to smell explosives, drugs, and other stuff. Same way, canines can also be trained to smell a person and alert them of their medical condition. So the what canines are smelling? So canines smell volatile organic compound. Um, these volatile organic compounds, VOCs, are byproduct of metabolic pathways that are altered by the disease. And most common sample types that these VOCs are emitted from are breath, urine, and sweat. And more recently, breath-based diagnostics are gaining much attention because of its non-invasive way of monitoring. So our approach is to find, so basically we know that dogs can be trained, they can detect or alert a person of their medical condition. So our big question is what are those VOCs that canines are smelling? And once we know what those VOCs are, can we make a nanomaterial-based sensor array system to detect those VOCs? So the research is two parts. First is biomarker discovery, where we use gas chromatograph mass spectrometer with machine learning to identify those biosignature of VOCs. And then second part is develop a portable nanosensor array to detect those VOCs. So how did I come across this work and why I got interested is about a decade ago, I was, uh, my background is, today. So my background is in sensors, and I was trying to search online to see what kind of sensors we can develop to help pets, right? Or to monitor pets or more pets' health. So while searching for that, I came across an article about diabetes alert dogs. And I read that article. Fortunately, that article was published by two local authors. One was a dog trainer, and one was from Eli Lilly, an endocrinologist. And that article was talking about how dogs can be trained to alert type 1 diabetes patients of their hypoglycemic event. Now, hypoglycemia, as you know, like if somebody experience, they can faint and even die within 5 minutes. So hypoglycemia is a very dangerous, you know, and subjects or patients who suffer from that, they are always, you know, very cautious about it. You know, they do not participate in sports. Uh, they they eat a lot of sugar, so you know there is other disease that come across as a byproduct of those control. So we wanted to see, basically I met with those authors and found like how they are training the dogs, what sample types they use. So I learned a lot about dog training from them. And then as an engineer, I was like, okay, so if dogs are able to distinguish two samples, one from normal and one from hypoglycemia, there should be some unique molecules present in those sample types that dogs may be smelling. And that's where our journey started to find those molecules and then develop sensors for the same. So uh, that project then, was we submitted a grant to NSF. We got NSF Smart and Connected Health funding to implement that project. We went to diabetes camp where there were hundreds of children who were type 1 diabetes. And my students, they they spent weeks in those camps, and as children were going from normal to hypo, while in a day they were able to collect samples from those children, and then we used to have a breath bag. We don't know if this pointer works, but so we used to fill the breath in a breath bag and transfer it to a vial and use the spinel fiber to run the sample through gas chromatograph mass spectrometer. And we were able to get the spectrum of VOC molecules and then run machine learning algorithm to find molecules that can distinguish normal versus hypo uh, with the least number of VOCs possible. And we were able to find 6 VOCs in the samples that can distinguish hypo versus normal with more than 98% accuracy. Then, once we Had that, we went next year again to the same camp. We collected samples. Now we blind tested the 6 VOCs within those samples, and we were able to get the accuracy of 93%. So it shows that the targeted non-target blind testing results were as accurate as the training set. So once we found that, we started developing the sensors. So we have two types of sensors. We have in-house sensor chip, and we have commercial sensor chip, where we can have a sensor array. In this case, we have 4 sensor elements. Each sensor element can be designed specific to a molecule, VOC of interest, and we can then make those integrate those chips into the electronics to get the signal from the sensor chips. So generally, to make the sensor chips, we use the lithography process, but the sensing element itself. Yeah, so the sensor elements were the sensor chip was fabricated using lithography process, but each sensor element, the material, the sensing material was coated either through electrospinning, where we can make nanofibers of nanocomposite materials that are sensitive to certain VOCs, or we can just spin coat those composites onto the sensor layer to make those sensor array system. The testing setup, we had both the simulated breath. So basically, we took some components of the breath and tried to mix and match to simulate the breath, and then tested those sensors using the setup shown on the left. And then the setup on the right was real Excel breath that sensors were exposed and tested in a testing chip. And here are some of the results. We made materials, composite materials using PVDF carbon black. We also used PI carbon black based materials. We used PI gold nanoparticles, and each material composite is distinct enough and also specific enough to send certain VOCs. For example, the PVDF carbon black was more sensitive to acetone compared to ethanol. We made the PI carbon black, which was more sensitive to nonanol, and we tested with different chains of nonanol, and we were able to find that it is more sensitive to just the nonanol, even if you have similar molecules. Then the PI, gold nanoparticle based sensing layer, was more sensitive to octanol and nonanol. So, and by combination of these materials, you can make a sensor array that can sense different VOCs. Especially when we are talking about sensors, we have to also make sure that not only they are responsive and sensitive enough, but they also can work like their shelf life. Because when you make polymer-based sensors, there is always a issue on degradation. So we did a degradation study to see how long this sensor can give you the similar response over time. We also evaluated the commercial sensors. These were made by Nanos. They are metal oxide-based sensors, and basically, we have the evaluation kit where we have the 2 sensor chips, and we did develop a calibration curve for acetone, isoprene, and other VOC database, and also evaluated the limit of detection for those VOCs. Finally, integration of the sensor chip into the device. So we made an in-house breathalyzer that is seen on the top here. And then we also worked with, this technology was also licensed by one of the company in California, Sports Industries. And we helped them to develop the prototype with the commercially available metal oxide sensors. Now we are launching the product in January. Uh, but before that, we are now at the clinical trials where the prototype is used with type 1 diabetes subjects to use it throughout the day and see if they can be used to alert them of their hypoglycemic or hyperglycemic events. So this project shows from the start to the end where we had an idea, got some research funding, then did the work, generated the IP, collaborated with the industry, and now taking it to the market. So then, based on the same concept, we have other projects. When COVID hit, we all felt the need for testing and the RT-PCR, though very accurate, there was bottleneck because of the process time for RT-PCR. So we wanted to see if we can detect COVID using breath, just VOCs in breath. And we were able to show that by just using 3 molecules in breath, we can detect COVID with 100% accuracy. And there were about 10 molecules in the breath that were changing compared to the healthy subject. And of those 10, a combination of 3 could give you 100% accuracy. Not only that, what we found was when these subjects who had COVID recovered, we again collected their breath and found that those VOC levels went back to the healthy controls that we had. So the figure on the right-hand side, the linear discriminant chart shows that the green one is the control and the blue is the recovered, whereas the orange and the red, the COVID-1 and COVID-2, are the COVID samples. Now we saw some distinction within the COVID sample, and we plotted them as COVID-1 and COVID-2, and it happened to be that the subjects who which were under COVID-1 had low symptoms compared to COVID-2 samples. So with that information, we submitted a proposal to DoD where they were seeking for to create a database for VOCs in healthy breath. So basically, they wanted the DoD wanted to have what is in the breath of a healthy subject, and we submitted the grant. We were one of the 3 who got funded in the nation to a national lab, and we were the only university that got funded for that proposed program. And we were able to develop now, like how you go, like you can. The idea is basically if you know what is present in a healthy breath or what are the concentrations of those VOCs in healthy breath, then any deviation could indicate a medical condition for those VOCs. So rest, I will just, we collected about close to 164 breast samples. And in that, we had 10 volunteers who gave their samples over time. So we not only collected samples of subjects like across the different demographic, but also same subject giving breath sample over a period of time and wanted to see how their breath profile changes. We could not see any distinction between the volunteers who gave their breath sample over time. and even across the cross-sectional samples, that means across subjects. That means we see that there is no unique pattern within an individual or across the individual, and that could correlate. Then that shows that there is a universal healthy breath baseline. We have a set of 64 VOCs that were present across the samples, and we have also identified their what we call the concentrations. We not not only trying to find VOCs across subjects, but also are there any confounding variables that can correlate to these VOCs? And we found that isoprene was lower in female compared to males. And when we dig more into those, the isoprene, we found that the isoprene is the byproduct of metabolic, the musculoskeletal metabolic process. So that shows that why we have the difference between the isoprene between males and females. Other than that, we could not find any other confounding variable that affects this the database that the VOC database that we have created, including the BMI uh, and age and other confounding variables. We also expose the healthy breath to our sensor systems and extracted several features from sensors to develop a algorithm that can show or make the sensor learn what a healthy breath looks like. And now we are working on spiking the healthy breath with other VOCs of interest to see if the sensor can respond to those and detect those VOCs. So more recently, as Rose Rusamano mentioned, that we also got a NIHR1 grant for cystic fibrosis and diagnosing for diagnosing pulmonary exacerbation in cystic fibrosis patient. So the reason, basically, the way we work is we go to the clinician, we try to find what the problem is. And once we find what the problem is, we try to see, okay, how we can solve what current technologies are being used and how we can solve those problems by means of new biomarker discovery and diagnosis. So we had a collaborator from Riley Hospital for Children, and they were looking for how to diagnose pulmonary exacerbation because current method is they use spirometer, and sometimes even with the spirometer they cannot tell whether the condition is through the pulmonary exacerbation or through other infections. And generally, these patients get prescribed antibiotic, and over time they get resistance to those antibiotic. So you can understand how big of a problem. I remember last year there was a shortage of antibiotic, and not only to help with the supply chain management of antibiotic, but also to avoid long-term resistance to those antibiotics. We need to reduce the prescription, and to reduce we need better diagnostic tools to avoid any misdiagnosis, for example, in terms of pulmonary exacerbation and viral or bacterial infection. So we wrote the grant to NIH, and we recently received the award, and we are working with clinician and other collaborators to help discover the biomarkers of pulmonary exacerbation and develop a device that can be used in the clinic or at home. Another project with that relates to more supply chain management and product authentication is we work with Crane. Crane Indiana. It's a DoD based naval base, right?
Surface Warfare Center.
Warfare center, yeah. So they were using X-rays to non-destructively analyze microchips and identify their authenticity or tamper, anti-tamper, programs. So we propose to see that can we tell by just smelling the chips where they were manufactured from and have somebody tampered them. And we were able to, so we just did a pilot study on that. And we had what we call different microcontrollers, similar type of microcontrollers but from different manufacturer. And the country of origin was different. And also different types of, not only the same microcontroller for different manufacturer, but different microelectronic chip too. And we found that there are 4 VOCs, 3 upregulated, 1 downregulated that can distinguish all 3 type of devices. So you can see the PCA plot here for all the 4 VOCs. You can easily distinguish the micro SD from the microcontroller manufactured by Texas or microcontroller manufactured by Microchip. And we also ran some blank samples to see if there are not coinciding with the VOCs of interest. Now, in supply chain, another application that we can use. I have not. We don't have this project, but this is just a concept. The freshness of fruit or vegetables is more important, and basically these can be the freshness can be detected using the ethylene that is emitted by the ripening of fruits, and we can make those gas sensors. Ethylene gas sensors are available to monitor the freshness in the warehouse, but can we make a low power passive ethylene sensor that can be just attached to a package or to a box that can monitor the freshness of the fruits inside that box. Now, what happens is one fruit, if they're rot, then the entire load can be destroyed. So we need to remove that box before the entire load that can get affected from the load. So uh, we we can make those freshness sensors using uh, thin oxide based nanoparticles. I did work during my postdoc on tin oxide based nanoparticles to make a capacitor, and the dielectric of those nanoparticles change when they are exposed to ethylene, and based on the change in dielectric property, you see the change in capacitance, and you can make use of the capacitance load in a uh, patch antennas that can change the frequency, which you can detect wirelessly for detecting those change in capacitance. In turn, detecting the change in ethylene. So yeah, if we can make the metal oxide based ethylene sensors that can be, because sometimes for these sensors to be reactive, they need heating element and a lot of power. But if you can make low power ethylene sensors, definitely we can make use of them in a passive sensing of ethylene for freshness detection at different supply chain, distribution centers. Other ongoing source. I'm good on time. So other project is Again, this was also inspired by an article published in Italy that there were dogs that were trained to smell urine to screen for prostate cancer. So I collaborated with a veterans hospital, which is like 5 minutes away from our lab, and we collected a urine sample and shown in a pilot study that there are molecules in urine that can correlate to urine headspace that can correlate to prostate cancer, and we wrote an American Cancer Society proposal and got awarded to develop a screening test for prostate cancer from VOCs in urine headspace. And not only that, uh, currently most of the VOC discovery happens in a benchtop instrument that is in the lab, GCMS, gas chromatograph mass spectrometer. But this project will also evaluate portable GCMS for detecting those VOCs that can be then used in a clinic for prostate cancer screening. So not only at home solution, but also solution for screening in clinics, and especially in the rural areas where uh, we can help early, you know, um, early screening of prostate cancer uh, in not only at home but also in clinics. Other project based on VOCs is a collaboration with Cornell University. The, pro, the, the researchers there approached us saying that, hey, there is this what you call the southern chain pouch rat is which are large rodents. They have a very keen sense of smell, and they can discriminate urinary VOCs to guide for social decision making. But they also can be trained, or they can also sniff bombs. So there should be some correlation between the VOCs in urine and also the VOCs in explosives. So we are working with them to identify the VOCs that are in urine that helps them make the social decision making, but then also see, once we find those VOCs, how they are related to the VOCs that are in explosives, and we found that they have similar structure. And this project is ongoing right now to understand the the differences and also similarities between the VOCs in urine that these African bush rat smells com- compared to what is in the explosive that they can also sniff out. Other projects that the institute or I collaborated with is on making nanofibers. So as you have in my previous slides, I showed the nanofibers that can be spun on a sensing layer to specifically sense certain VOCs. Same way we can make nanofibers for making carbon fiber reinforced polymers. Uh, so if you remember Titan, the body of the Titan that got submerged, um, the submarine was made from carbon reinforced polymer, right? Uh, so, and these CFRP, carbon fiber reinforced polymer structures, are made by putting carbon fiber plies, several of them, and laminating them together. And the main failure that happens, if you read a recent article that happened because of the delamination. So the main failure of those carbon fiber reinforced polymer is delamination. And what we did was, before even the article got published, we knew that there is this failure mechanism, and how we can improve this failure mechanism is by adding epoxy carbon nanotube embedded nanofibers in between those plies before you laminate, that can improve the strength by more than 25% and yield by more than 400%. So we had this technology back into 2020, and we started our company and received a NSF SDAR phase one. And now we are looking for more applications and phase two funding for this technology. One other area that we can use this nanofibers is in filters. Not only we can make this nanofibers, but we can attach these nanofibers with copper oxide nanoparticles that can then disable the viral particles within. So not only these active filters can reduce the number of particles or viral uh, particles, but also deactivate them upon contact using the electrospinning technique that we have developed in the lab. So that's about the research. I want to now talk about the Integrated Nanosystems Development Institute that Provost Sulsamano mentioned. We started this institute about 13 years ago, and with very limited resources, but from but what we did was we put together limited resources and and formed a group of faculty that works in the area of nanotechnology and applied for several instrumentation grant. You can see all these instruments were funded by NSF MRI, all the way from field emission scanning electron microscope to a recent IFM and nano IR, or to fund the XLD. Each equipment is unique of its own. For example, XRD, we have a high temperature stage that can go up to 2300 degrees centigrade, that is half the temperature of the sun. So not only you can do material analysis, but you can do you can get the material characteristics in situ by increasing the temperature and for applications such as in hypersonics. We have other fabrication and characterization equipment. So most of these were funded by external grant. We have industry advisory board. We work closely with industry partners in the region. And then we not only the research and equipment, but we also help bring the outreach part to the institute by established programs such as NSFRU. We've been running this program for 8 years. We bring in undergraduate students from limited research universities to come to campus to work with faculty on various projects. We also have NSF RET, which is now funded for almost 8 years. And we bring in high school teachers from socioeconomic disadvantaged school districts and place them with faculty to work on summer project. But the outcome is not just the research experience, but they also create teaching modules that they can go back and take it to their classroom. We had NSF NSF iTest program, which is innovative teaching experience for students and teachers. And we got high school students to the campus for hands-on experience and helped several teachers to create course modules for their classroom. Some of the students from my lab, you can see that not only the PhDs are always like, they go to postdoc and good academic or industry jobs, but we have master students working in the lab also achieve and go and work for industries. So these are some examples of students from the lab. Uh, so Sophia Zankari was my high school student. She worked in my lab during the summer, then took a break before joining the college. She worked for a lab for a year in my lab and was able to publish and then go to MIT after finishing that 1 year experience. So in acknowledgement, these are my current students and postdocs in the lab, including undergraduate students. You can see that I just don't have, students from one major. We have, it's a very interdisciplinary group, all the way from chemistry to electrical engineering to computer science to pre-pharma to biology and other subjects. And these are some of the units on campus or the funding agencies that have supported the work. One thing I would say that, as Provost Rusamano mentioned, it's not only the external and internal support that helped me to be here, but also Provost Rusamano support and encouragement made me today stand in front of you and give this talk and all the accomplishments that I had over the past decade. So I really thank you, David Rusmano. So, um, you know, so old memories. Thank you so much for all the support and encouragement. It's very important. It's just not the money, it's just not the projects, but you need leadership and that the support from that leadership can definitely go a long way than your research funding or your small projects. So thank you so much for the support. With that, I'll conclude my talk. Thank you so much. If you have any questions, I'm here to answer. Yes.
What's been the feedback from local industry, like a lot of the uh, projects of the research that you've done? Has there been a lot of interested parties, or have you found that you effectively aligned what you're doing to match with the industries asking for?
So so there is a lot of interest in locals, in developing tools for diagnosis, for prosthetics, there is always interest, and there are faculty members in the institute that are working on those projects. My project I started, as I said, from my passion to help, non-invasive health monitoring systems that can reach to underserved and socioeconomically disadvantaged population. So the way I work is I could have had my own startup in the medical field, but I have one on the nanofiber based systems. But I do collaborate closely with the industry who can support the medical research to take it to the market, like Sponge, who are in health, fitness, but slowly trying to see the area of medical field and how they can help in the medical. Online.
So Maggie, can you speak a little bit more to some of the DoD work, right? Because they were interested in really kind of a catalog, if you will, of signatures. Can you speak it to any more detail?
So the DoD project, yes. See, after COVID, even before COVID, I think in DoD, the warfighter preparedness is a big program. And they wanted to, so basically, they, like for Air Force, we have this, fighter jets that cost millions and millions of dollars, and they want to make sure that our white war fighters are prepared and they're uh, fatigue free. Sometimes fatigue is a big problem, and you cannot monitor those conditions just by non-invasively in warfighter. So they wanted to create. So basically, the idea is that how quickly you can find a medical health condition by non-invasively detecting those conditions. And they have been spending a lot of efforts on sweat-based monitoring. So they have sweat sensors that can monitor not only the electrolyte-based electrolytes and other analytes in the sweat, but also now VOCs. So the volatile organic compounds can give much more information in terms of your metabolic process that is going on in the body compared to other methods. So that's what.
Yeah, I mean, it sounds like a rich domain for discovery and application.
Yeah, like how you can go, like we have this every year visit to a clinic where they do the blood test. And what is the blood test? The complete metabolic panel, the blood count, so, and in that, basically they see for various parameters and the range. Same way, if we can have what is present in the breath, what is the range of healthy, and any deviation from the healthy, we can find what medical conditions they may have, and have the work. For example, I can tell, uh, you know, we have many sensors to sense chemicals and biothreats, right? On the gear when PP, like protective gear. But we don't have anything to show if somebody has exposed until the symptoms starts appearing. So can we have a sensor or a system that can detect if somebody got exposed and your body is reacting? You don't have the symptoms yet, but before you went into the situation of emergency, you can detect and take care of the warfighter.
So like pre-markers or something.
Yes. So your body is reacting to whatever
Yeah.
you're exposed
Did
to.
not show the symptoms till 48 hours later, so it's
Yeah.
like that. It looks like there were no questions online, which always means you answered everyone's questions. So just never posted them. So I'm going to give our speaker a big round of applause again.